Okay, well, we'll go ahead and get started. I'd like to welcome all of you again to one of our webinars. This is great to see all of you guys. And I think we'll just start it off with Rebecca's talk. That way we spend most of our time there. And then at the end, again, we'll open it up if you guys want to talk about your ongoing research, some problems you may be running into, or some cool findings that you have. And then also I'm going to show you, I'd like some of your feedback. We have a mock-up. They kind of like some of your ideas on what we're drafting together. And then if Dawn has any other kinds of questions you might want as we get going. So with that, I'm going to go ahead and turn off my webcam, turn over to Rebecca. Rebecca, I'll just let you introduce yourself to everyone. Looks like most everyone we have seen before. Celeste is new, but you can get her up to speed with who you are. So I'll go ahead and shut me off, and you can go for it, Rebecca.
Okay, I'm a second-year student in Jorge Dubkowski's lab at the University of California, Davis. So I see there's quite a Davis contingent here today, so that's always nice. So I'll just talk a little bit about my project. It's not specifically funded by TCAP, but a lot of projects in our lab are being supported by the TCAP program, and I'm just trying to be involved as a student in the plant breeding training network. So I'm going to talk a little bit about my project today, kind of give you an overview on vernalization and photoperiod in cereals, and then specifically talk about the genes that I'm working on. If you have any problems hearing me or I need to slow down or anything, just let me know. So just to give you kind of an overview for those of you who are not familiar with wheat production in California, they produced over 700,000 acres last year, which had a value of about 230 million. And some of the uses for those 700,000 acres, some of the uses of the wheat that we produced, the common wheat was mostly used for bread flour and general purpose. And depending on the price and the quality of the grain, some of that does go towards animal feed. And then the durum wheat is mostly used for pasta products, and there's quite a bit of export to Italy. So just a little bit about our lab. We have quite a few members of our lab, 8 postdocs, 6 visiting students, 5 PhD students, 4 lab techs, 5 undergrads currently, and a field manager, which I didn't put on here. And all of that is covered by 1 PI, and that's Jorge. So about 30 people, and we're looking to hire 2 more. I'm not sure who's clicking through the presentation there, but try to keep it on the same page here.
Yeah, this is Dina. So let Rebecca move to the slide. And I'm also hearing some background noise, so if you have some conversations going on, go ahead and mute your mic so we don't hear that coming through. Okay, sorry, go ahead, Rebecca.
So we are looking to hire, I think, 2 more postdocs for the TCAP project, so we're quite a big group. And we cover traits all the way from grain protein content to disease to flowering time, and we're starting to get into drought as well. So just kind of an overview of the flowering pathway. It can be kind of complicated, and there are some points that we're not entirely clear on yet. But overall, there are 2 main areas, one being the photoperiod pathway, and the other being the vernalization pathway. And so just a basic idea of how that works. You have PPD, which is a photoperiod sensing gene, which then moves into constants, which upregulates FT. FT is also repressed by VRN2 prior to vernalization here. And FT is, when it's expressed, it helps to upregulate VRN1, which is a meristem identity gene, which will eventually push into flowering. and we're not quite clear yet which, either VRN2 or VRN1, which of these two is actually sensing vernalization. When we do vernalize the plants, we see a downregulation of VRN2 and an upregulation of VRN1, but it's not quite clear who's moderating who there, or if there's another party involved. VRN2 is also down-regulated by short days. So we believe that there might be another repressor here. The CARG box is, we believe, primarily where VRN2 is interacting and repressing for the vernalization part of the pathway. And we think there's a second repressor maybe in the second VRN box, as we've called it, which is sensing the, sorry, I've got this backwards. So VRN box is for VRN2, and the CARG box is sensing that short day repressor. So, sorry, my computer's lagging a little bit here. So this is just kind of showing the components that are considered to be part of the vernalization pathway. And FT is actually observed to interact with another gene called FDL2, which will actually interact with the VRN1 gene. So it's been recorded that they believe there are at least 3 major gene pairs responsible for this winter versus spring growth habit as far back as 1971 with Takahashi and Yasuda with this paper. And so the genes that they discuss in their paper do actually correlate to VRN1, VRN2, and VRN3, which we know of now. And so because of the strong epistatic interactions that we can observe in different populations between these different genes, we do believe that they are part of the same pathway. So just a brief little introduction to each of these major genes. VRN1 is homologous to Apatala1, which has several paralogs in Arabidopsis, including cauliflower and fruitful. And when you see RNAi of this AP1 in wheat, oh, my arrow is not coming down here. There we go. When you see the RNAi of the AP1 gene, you do see kind of a stunting and a delayed flowering type phenotype here, whereas wild type has completed the flowering process here. And the dominant VRN1 gene, we usually see deletions or insertions, large retroelements, in the promoter or the intron 1, which then leads to an upregulation of the VRN1 as it's no longer repressed by some of these other players in the game. So then VRN2, as I said before, during vernalization, you see a downregulation of VRN2 and then a subsequent upregulation of VRN1. And then the recessive version of VRN2, where that's deleted, is where you have the early flowering phenotype. And we see there are actually 2 genes at this locus, CCCT1 and 2. And when those are deleted, or there's some kind of substitution in some of the main amino acids in the CCT domain, you see this early flowering phenotype in the recessive VRN2. And then with VRN3, otherwise known as FT, which is described as the florogen in Arabidopsis, when we see a dominant version, allele of the FT, we see this early flowering phenotype, as you can see here. And the wild type is much more delayed. And so this florogen is a small gene, a small protein that is part of the RKIP family in mammals, a kinase inhibitor protein. And so here, the dominant VRN3 is the insertion of a transposon, or in the case of barley, we see several SNPs that we believe are are linked to this early phenotype. And you do see a higher expression level of FT at this point. Come on, there we go. So then the photoperiod pathway mainly involves the PPD gene and constants. And so if you see an insensitive copy of PPD, then you'll no longer see the differences between long day, or you'll see a reduced effect of long day and short day on flowering. There's also this, which this is a little bit less explored, but there's this group of HAP proteins, which we believe are competing between constants and VRN2 for binding to FT, and we believe that they may have some other role in sensing different environmental components before determining whether or not the plant is going to go through and complete flowering. So here we just have a little bit of a diagram that's describing the different seasons. You can see here the constant expression in long days in fall, and then as you have shorter days in winter, the activation of constants is shown to be occurring when it's expressed during the light, which occurs during these long days here in fall, and then again in spring and summer, specifically in summer there. And you can see this overlap of the temperature here. We've got kind of the trend of the temperature going down in the winter here and then increasing. And you can see that VRN2 with the shorter days and also with the decreased temperature is decreasing in expression levels, while VRN1 starts to slowly increase, and then as VRN3 comes on, it increases even farther. And so we see the transition from a vegetative apex into the double ridge stage and then finally progressing into flowering here. Okay, so then my work is mostly on FT, characterizing different alleles of FT. I've also done a little bit of work, some kind of side projects on VRN1 and those different repressor sites in the promoter. But mainly my thesis work will be working on FT. And so again, in wheat, we see the dominant VRN3 has this insertion in the promoter with a retroelement insertion, which we believe is interrupting some kind of regulation site there and causing an upregulation of FT. And then in barley, we actually have several SNPs in the promoter, but we believe the SNPs that are in the intron are associated, they've been previously associated with the early flowering phenotype in the dominant VRN3 and the later flowering phenotype in the wild type or the recessive VRN3 allele. So I've kind of got three different directions that I'm going with characterizing the effect of FT in Triticeae. And so with barley, I'm trying to characterize some of the natural variation that we see in those promoter and intron regions. We have nine SNPs that we've identified that are linked in the promoter, and then 2 that are identified in the intron. And so I'll discuss a few populations that I've worked on looking at the effect in different backgrounds with those different promoters and introns. And then in wheat FT, this is more of a future project. I haven't really started it into this direction yet, but I will be characterizing the previously described hope allele of FT, which does have that retroelement insertion in the promoter. And we've done a series of back crosses into different varieties of tetraploid wheat, and we'll be putting those into a field environment to characterize the differences in the different backgrounds there. And then finally, there's a reverse genetics kind of approach called tilling, which I'll describe a little bit later for those of you who haven't heard of it. And so basically I'm trying to develop knockout or knockdown lines by chemical mutagenesis and then back crossing those to reduce the background mutation and then crossing them together to create a complete knockout line. So this slide is a little bit complex, but it's just intended to kind of give you an idea of the different haplotypes of the promoter and the intron in barley FT. So this yellow box here is describing the early promoter type, the SNPs that have been associated with the early phenotype. And then this blue box at the bottom is the later phenotype. And then again in the intron we have late shown in brown and then green indicating early. And so you can see that with the VRN1, VRN2, and then VRN3, which is describing the state of these promoter and intron effects, it gets quite complicated in the information that we have to sort through when we try to determine the effects. So we've made some various crosses with both insensitive and sensitive alleles of PPD, and then looked at those in the greenhouse in different populations under long day conditions to see the effect of the promoter and the intron. And so in previous work we've actually described, there's a PNAS paper in 2006 where they did associate VRN3, or barley VRN3, with the FT Arabidopsis phenotype, and so they did describe quite a few of these haplotypes. But we have a novel one that was not described in that paper, which has the late promoter and then the early intron combination, which has allowed us to try to dissect that promoter effect a little bit better. So here you can see this is just kind of like I said, it's very complicated. The different populations are very complicated in all their combinations of vernalization genes and photoperiod genes. So we've tried to break these down a little bit. And so this first population with BGS213 and I330. BGS213 has the dominant VRN3 gene, which is primarily what the 2006 paper was using to show that association between VRN3 and FT. And we see there's a very strong interaction between VRN1 and FT here in a PPD-sensitive background, and we see in the recessive VRN1 background, we're able to see a difference of about 5.6 days, about 6 days, where the early promoter with a fixed intron background, the early promoter is able to increase the flowering time, or decrease the flowering time by about 6 days. But we don't see a significant effect when you get to the dominant VRN1 because it's already pushing itself through to earlier flowering. If we look at spontaneum crossed with a line called E878, spontaneum has that recessive VRN1, but we believe it's a little bit stronger than the VRN1 allele that we see in I330. And if you see here, there's again a very strong interaction between VRN1 and FT. And we broke this population down into both sensitive and insensitive lines and looked at some F3 families. And we did still see the effect even in the insensitive PPD background. We see about 40 days difference, 40 to 50 days difference in the recessive VRN1 background, the sort of wild type VRN1 background. And only about 5 to 10 days difference when you have that dominant VRN1 pushing through. And this is with a fixed late intron type. So we've crossed spontaneum with a yet stronger version of the VRN1 allele, and we see no significant effect between the interaction of VRN1 and FT, and then likewise we see no significant effect of VRN3 in these populations. In a similar cross here with spontaneum and Morex, which Morex is believed to have the same allele as BGS213, which was shown here to have a very strong effect on flowering time, but it's not able to overcome the VRN1 allele in this case. And so we believe that there may be an additional factor that is moderating these interactions that we are unaware of, or something that's closely linked to RFT alleles that we don't understand yet. So this is kind of a complicated slide as well. But when we were actually looking at the FT allele and trying to clone this gene, or sorry, looking at the FT gene and trying to clone it, we did have several gaps in our back clones that we weren't able to complete because of significant stretches of retroelements. And so we are looking by collinearity, comparative genomics. In the PNAS paper, they compared the barley genome to the rice genome. And so they didn't see any additional predicted genes that were unaccounted for here that they felt would be associated with this early phenotype. You can see that here FT is duplicated in rice, but we didn't see any additional genes here. But when you look at brachy, which is significantly closer in the evolutionary pathway to barley, we did see a few genes here that we're starting to explore, but we're not completely convinced that they are having an effect on the flowering and complicating these populations. So we're currently exploring between the Morix allele and the PGS213 allele. We're trying to look for differences there to see why, in some cases, BGS213 is able to overcome strong alleles of VRN1, but Morix is not. And so far we've gone just upstream of FT. There's about 1.3 kb region of promoter, and then you run into just kilobases and kilobases of retroelements. So we don't believe there's anything upstream anywhere close to FT that's moderating this effect. But we do see there's a predicted gene in barley about 6.6 kb downstream from FT, and we were trying to see if we can see that gene also in Brachypodium to see if it may be causing the effects that we're seeing and complicating our populations. So just a quick discussion about the wheat FT allele. The HOPE allele, as we call it, does have that promoter insertion in VRN3, which leads to a dominant phenotype. And when we transformed this into a winter line of wheat called Jagger, we did see significantly increased or significantly earlier lines that were linked to that insertion. And so this allele is actually out of a collection of 19 tetraploid spring wheats, 29 hexaploid winter wheats, and 77 hexaploid spring wheats. We did not find this allele in any of that collection. And so we believe that we have a potential here to move this into commercial lines and utilize this genetic diversity. So now if we can determine the effect of it, it'll be a lot more useful. And unfortunately, we've seen, depending on the environment and the background, we've seen varied effects. So there was an experiment in California where the allele was not able to overcome the background and did not advance the flowering time. But the experiments, which have been repeated twice in Argentina, it was able to confer this early flowering phenotype. So now we've back-crossed it into 5 different tetraploid lines, and it's intended to be included as part of the field trials in fall of 2011 so that we can see if we can characterize some of the differences of effects in different backgrounds. So finally, the final part of the approach of my thesis here would be tilling, which is known as targeting induced local lesions in genomes. And this is a reverse genetics technique where we use EMF chemical immunogenesis to mutate the genome, and then we do a large-scale screening of our population of about 1,500 individuals. And essentially, we can find a knockout gene or at least some kind of significant amino acid substitution in a conserved region of the gene in a majority of the regions that we look at. And so there are 2 approaches to screen these populations, these mutant populations. And the first one is by Cell 1 Digest. So Cell 1 is a celery extract, which we believe includes this enzyme called Cell 1. And Cell 1 is responsible for recognizing heteroduplexes between DNA where you have a base pair mismatch, and so it almost gives a little bump on the DNA strand, which is recognized by this enzyme and clipped. And then when we run it out on a gel, we see those digested bands. And we're finding that there's a use of next-generation sequencing in screening these populations as well. We're doing various pooling schemes, which are described in Sci 2011, where we do deep sequencing of these different pools and then look for the mutations and then deconvolve to find out the single individual that has these mutations that we can use in our target genes. So far, I have successfully identified a splice site mutation in FT in the A copy of tetraploid wheat, but I've only been able to identify amino acid substitutions in FT in the B genome and also in the pyrrologous gene FT2 in the A and B copies. I also only have amino acid substitutions. So the next step, aside from further screening of the hexaploid to look for a more significant, more deleterious mutation in the FTB genome. Aside from that, the next step is to cross these homologous mutations and create a complete null. And the reason that we have to do several back cross generations before completing these crosses and combining the FTA and FTB alleles, for example, is that we have a significant background mutation because of the EMS mutagenesis process. And so in a tetraploid line, for example, you can have as many as 260,000 mutations in a single individual. And then in hexaploid, you can see even an even greater number, almost double that, because of the polyploid nature of these organisms and their tolerance of that mutation. So, like I said, back cross generations are necessary to reduce that background mutation before crossing them together in order to ensure that the phenotype that we're seeing is related to the mutation that we think we're looking at. And the more subtle the phenotype that you have, say a couple days difference in flowering time, more back crossing is necessary to be able to see this mutation or this phenotype. So in summary, in my 2 years of work on my thesis, the future directions I have here on barley FT would be to complete additional populations needed to determine the effects of the intron or the promoter SNPs. And if there is an additional gene that's moderating these epistatic interactions, we need to open up some of these populations and see if we can determine what that is. And then in wheat FT, we need to continue multiplying seed for this upcoming field trial of the back crosses of the dominant FT allele in tetraploid varieties. And then finally, in tilling of the FT paralogs, we're currently in the stage of back crossing and combining these mutants for future experiments. And then we will be doing populations of those as well to see what the effects are there. So finally, I'd just like to acknowledge a couple people in our lab. Of course, Jorge Dubkovsky, my professor, my major professor, and some postdoc mentors, Asaf Distefeld, who has since moved on to become a professor in Israel, as well as Stephen Pierce, who is a postdoc in our lab now. They've provided me great guidance in my work. And there are several Dubkovsky lab, of course, all of the Dubkovsky lab members, but specifically some that have come before, including Lu-Ling Yan, who was one of the major contributors to that PNAS paper that previously described the allelic variation of FT, and also our collaboration with the Komai lab in developing the next generation sequencing tilling platform for polyploid wheat. And of course, all of the gracious and generous funding sources that have helped with our project, including the USDA and several UC Davis-based fellowships. So if anyone has any questions, I'd be happy
go ahead and give her a
to
round of applause,
explain,
and
I'm sure this is all about as clear as mud.
So you can applause or, yep, okay, you guys are finding it. Excellent. And it looks like Josh has a question, so we'll turn the mic to you. Go ahead. Or maybe not. Maybe you just rose your hand.
Oh, sorry. Can you hear me now?
Yep, hear you great.
I was curious if you could talk about maybe some field applications for the work that you've been doing, if they have applications to make faster varieties, or what the end product that you might see out of this.
Well, as I'm sure many of you are familiar with, a lot of the TCAP program is addressing some various environmental challenges that we have in wheat production, including climate change. So understanding these different alleles of FT in barley and wheat can allow us to sort of design these cultivars to adapt them to specific climates,
has a question.
even while the climates are changing around us.
All right. Rebecca, have you genotyped any of the parent lines that Jorge uses in his wheat breeding program, so that, you know, breeders have been taking flowering date for a long time as phenotypic data, but now do you see that your team is looking at flowering in a more specific way because they can genotype these specific genes and maybe be more predictive about what the possible progeny genotypes and their flowering responses will be?
As far as I'm familiar with it, I know that they do tend to genotype VRN1 as being the major player there in flowering time. I'm not familiar with any specific applications yet in using FT, but as we understand this gene a little bit better, I'm sure that will be a player. As I was alluding to before, some spring lines will tend to have the dominant VRN1 and the recessive VRN2 phenotypes, so that they flower without that vernalization requirement. And then in your winter lines, you'll tend to see the recessive VRN1 and the dominant VRN2 allele.
Hey, can you hear me?
Yep. So Liana,
okay. So I was wondering, do you have any idea about the mechanism that occurs when you have a vernalization-sensitive plant that then kind of just puts up a few spikes here and there? Do you know what I'm talking about? So, I mean, you can see sometimes a plant that's very clearly requires vernalization, but will maybe put up just like a spike or two. Exactly, without vernalization. Yeah.
Without vernalization, but you mean when that plant is also vernalized, it would produce more. So we believe that that's, I was mentioning before, we have kind of a weaker allele of VRN1. One of George's favorite ways of describing it is as VRN1 being a kind of a lock. And that lock is of various strengths depending on which allele of VRN1 that you have. Maybe you have some SNP there that predisposes it to being more similar to the dominant earlier flowering phenotype. So if you have a stronger allele of VRN1, you probably won't see any spikes coming out without vernalization. But the weaker alleles, they may take a little bit longer without vernalization, but they will eventually flower on their own.
Does that answer your question, Leanna? Or
increase very gradually.
Oh, yeah, that sounds
cool. Other questions for Rebecca? So Rebecca, this is Dina. I'm kind of following up on Don. I see he has a question too. So do you kind of, I'm trying to envision the big picture here. So could it be possible then that breeders could screen their germplasm for flowering dates without having to always grow them all out in the field and wait for the season and see what really happened?
Yes, of course. So the idea behind this project is to characterize those different alleles and kind of give an idea. Maybe this allele will confer maybe a week difference. Maybe this one confers 40 days difference. And then we would have markers, which I believe for VRN1 and some other genes, George does have some of those markers developed and listed on his website for breeders to use in their germplasm and screen their parents before combining them. But with this further characterization of the FT alleles, we may be able to provide a few extra tools that they can also combine. And knowing what backgrounds to incorporate those alleles into
okay, I see. Thanks.
May make it a little easier for
Don, go ahead with your
to
question.
really narrow down what effect they expect to see.
I must not have been paying enough attention when you talked about production in California. Is wheat always grown under irrigation in California?
I'm not sure specifically about that. I believe there are some areas that are rain-fed. As you can see, I can get back up to the map here. Some areas of Southern California where you wouldn't have access to irrigation throughout the whole growing season, those are most likely rain-fed, but then there are some other areas in Northern California that have more access to water and irrigation throughout the growing season, where irrigation would be more likely incorporated. This year we had quite a bit of rainfall even late in the season.
With our in Nebraska, with our irrigation resources that are mostly used for soybean and for corn, there's just a lot of emphasis on the timing and having it fit in with flower development and its relationship to a yield response from that water. So I was just wondering if this might be another application of knowing the genotype and knowing when the plant's going to be going through different developmental stages and how that might help irrigators in California.
Well, of course, knowing the amount of time that it's going to take to develop your grain, and if you can decrease that time to match when you're going to have water available, that would certainly be a great application for this knowledge. But yeah, I think you make a good point there of being able to cater your
cool. Any other questions for Rebecca?
To the resources that you have available to produce those.
All right, looks great. Thanks, Rebecca. I'm going to go ahead and switch gears a little bit. I want to stop sharing your presentation and show a PowerPoint I have on my desktop. I didn't get it saved as a PDF, so I'll just go directly to my desktop here. Just a second. Actually, I'll stop recording because people won't need